Hello everyone, and welcome to our first note session for chemistry. Anytime that we do notes, you'll notice at the top of the page are some boxes that have different vocabulary terms, which we will be defining together. Then you'll be able to reference it throughout the week to help with your assignments. So let's go ahead and jump in to define chemistry. Great place to start. Chemistry is the study of matter. And matter is anything that has mass and takes up space. And if you think about really anything that's tangible, that's matter, whether it's atoms or compounds or mixtures, that would be what we're studying, matter in chemistry. There are 5 different branches for us to study matter, and those 5 branches each study a slightly different area. So first we have organic chemistry, which is the study of mostly non-metal compounds through the element carbon at its center. Inorganic, on the other hand, centers around metallic compounds. Biochemistry has to do with bio, meaning life, life reactions. Think like metabolism or photosynthesis, reactions that happen in living things. Analytical chemists study what makes up a sample of matter, and they are looking at the composition of the matter. And lastly, physical chemists study reactions and ways that the observation relates to internal structure. Whenever we're studying any science, we're going to be doing a lot of things with experimentation, and experimentation centers around the scientific method. I'm going to loosely define the scientific method as the process of human curiosity. We start the scientific process very casually at a really young age through some sort of experimentation, and then we collect some data and we make a conclusion about what we've observed and move on with life. Maybe we make up another experiment to verify our findings, or move on to something else that sparks our interest. That is loosely the scientific method. When we're working with an experiment, we want what's called a controlled experiment, meaning we're only changing one variable at a time. Let's talk a little bit about variables for a moment within an experiment. First, you want to have an independent variable, and only one in a controlled experiment, because your independent variable is what the experimenter changes or manipulates in order to figure out the response. That leads me to the next variable, the dependent variable, which responds to what has been changed or manipulated. Anything else in the experiment that is held constant is a control. Held the same. So again, we want our experiment to be controlled, meaning it only has one independent variable. Let's go ahead and jot that down, that a controlled experiment only has one independent variable, one thing that is changed or manipulated. Otherwise, we don't know what the response is related to. Let's take a look at our example here to apply the scientific method and its steps. You'll see the 6 steps listed: observation, question, purpose, experiment, results, conclusion. Go ahead and pause the video and work through this on your own, trying to match the example with the parts. Okay. I'm going to read through this and pick each part out as we go. I'm not going to write it, I'm just going to tell you these verbally, but you write them down on your page. So the example says, you notice that adding acid to a chemical solution can cause color change. This is our observation. You notice that adding acid to a solution can cause color change. There's the observation. Our question is then, well, which compounds change color? You're trying to figure out which compounds change color. You could put that right there as the question. Then you decide, okay, I'm going to make this a little bit more specific now, finding a purpose within my experiment. So it shows that we are going to be looking at 3 specific compounds: sodium chloride, silver nitrate, and magnesium acetate. By the way, I'm writing right now the chemical formulas. I will teach you how to do this later in the semester. In the meantime, let's continue with this problem. Our purpose now is to identify reactors with acid, specifically NaCl, AgNO3, or MgC2H3O2. Which of these solutions will react? That's our purpose. The experiment then is where we start finding kind of the steps. We're going to add a drop of acid to each solution. You'll probably observe what happens, record it in a data table, and that leads us to our results. What is our result? We want to record, did it change color or not when the acid was added? Usually you'll have your results in a data table. I'm going to sketch one real quickly here, putting the solutions on the side, just listing them out. And then we'll write the response, color change, at the top. Using our table, then we can record yes or no, or we could record what color it was before and after. For this, I'm just going to put yes and no, and I'm going to make up this data because I have not done these reactions. But there is an example of what your data table might look like with the result. You are telling me, did the color change happen or not? That's the result that you'll be giving. Then the conclusion is a description in paragraph form, telling which solutions changed color, maybe an explanation of why that happened, or why other ones did not change color, linking it to potentially the internal structure of the compounds themselves. Next, we have an identification of the variables in the controlled experiment. Let's break this down. Independent variable is what the experimenter changes or manipulates. In this example, we have three options of the solution. So that would be our changed or independent variable. The dependent variable, the response, would be the color change. And notice, independent and dependent variable are used in the data table. What did we change? What was the response? Lastly, our control is anything that we kept constant, like the glassware that we're using. We want to use the same type of test tube for each solution. The amount of acid, it says we're adding 1 drop of acid to each. We also want to keep, it says, the volume of each solution constant. So those are our controlled variables. Moving down here, it says, what measurements might have been used when collecting data? Well, let's go back up to our vocabulary section to discuss a little bit about measurements. When we're talking about different types of measurements, we give them a label called a base unit. So we're going to describe our base unit as a label given to identify different types of measurements. And you probably are very familiar with these different base units, but we're going to write them down for reference. Length, we use meter, not foot or inch, but meter within the scientific community. Mass, not pounds, but gram. Volume, we use liters. Temperatures, degrees Celsius or Kelvin. More to come on Kelvin, but that scale, notice it doesn't have a degree sign. It's just Kelvin, named after the scientist who created that concept. And time is seconds. Lastly, energy is joule. Now, what if you have a lot of base units or just a fraction of one base unit? How can we transform our unit to give us the proper amount? We can use what's called a metric prefix by transforming the value by a factor of 10. So it transforms our unit by a factor of 10, giving us either a larger amount or a smaller amount. For example, little k here stands for kilo, and you've probably heard of a kilometer. That's a kilometer. Kilo attached to meter means that we have 1,000 meters, or 10 to the third power meters. Kilo is 1,000. Hecto is 100. Deca, DA, is 10. Think like a decade. Base is just 1. If I have a meter, that is implied it's just 1 meter. Little d is deci. Now we're getting into fractions. 1/10, 10 to the negative 1 power. Centi, if you think even just like a penny, it's a cent. 1/100th of a dollar. And milli is 1/1000th. 10 to the negative third power. You can use any metric prefix with any base unit to transform it by a factor of 10, larger or smaller than 1. A mnemonic device to help you remember the order of these is King Henry died by drinking chocolate milk. I didn't come up with it, but it does help you remember. Let's revisit our last two questions here. What measurements might have been used when collecting data? Well, our response that we're recording is color change, which isn't really one of the measurement types. However, we do want to keep our volume of each solution consistent, so we'll definitely need to use milliliters for a quantitative value. Our actual dependent variable response would be recorded as a qualitative value. Maybe the color or yes or no, just a qualitative response. But milliliters will be helpful to continue keeping that solution amount consistent. That's a volume. And lastly, to which branch of chemistry does this experiment belong? We talked about how reactions tell us a little bit about the internal structure of it. So that would mean this is related to physical chemistry. In your workbook to the Design Your Own Experiment practice page, you'll see these 5 different scenarios that are given. They're each observations of true observations that could occur. And then you are going to write your own experimental design for 2 of them, including a question, a purpose, a description of your experiment, a possible data table, list out the variables, figure out what measurements might be useful, and classify your experiment into one of the branches of chemistry. So let's say I choose the first observation. I need to come up with or follow that process of curiosity. Here's the observation. When water has a certain amount of calcium or magnesium in it, soap bubbles will no longer be produced in water. This is called hard water. That's observation A. I'm going to go to the next page where I have this box and I circle observation A. And now I'm going to create a question. Maybe you're asking how much calcium can be in a sample before there's no soap left, soap bubbles. Or can there be a little bit and still have soap bubbles? You can come up with your own question. Then come up with a specific purpose, a brief description of your experiment. It does not need to be lengthy. In our example, it was add 1 drop of acid to each solution. Then a simple data table. List your variables, what measurements might have been used, and a branch of chemistry it's related to. You'll do that for 2 of the observations on the previous page. The other examples are B, certain objects will sink while others will float even if they're the same size. C, diet is important for physical activity. Some foods make you lethargic while others fuel your body. D, your hair feels silkier using one brand of shampoo over another. And E, some metals are magnetic but others are not. You choose 2 of these and fill out the tables on the next page.